All right, so this is the 14th meme seminar given by Dr. Lafi Aldakak.
Actually, doctor. I have
Oh
two
yeah,
doctor
right.
degrees
Dr.
now.
Lafi Aldakak. So Lafi started meme with me and Julie in 2014, and he had already had an MD by then from Damascus University in Syria. After meme, he did his Ph.D. in the Institute of Evolutionary Medicine at the University of Zurich in Switzerland, where he is now currently a postdoc research assistant. And he will be talking to us about evolutionary medicine, I believe. So
Sure.
go for it.
Okay. So I'll share my screen. So thanks, Charles, for the invitation for organizing this seminar, and thanks everyone for showing up. I'll tell you about the work I did in my PhD on the evolutionary regulation of immune responses. So just an overview of my meme experience as Charles has said, after I finished my MD in Damascus, I joined meme, where I started actually in Groningen, where I had a very nice time. But after the gray, rainy winter in Groningen, I went to Montpellier. And as I saw the sun there, I was like, just give me more of this. So I was the only one to stay for 2 semesters from my cohort in Montpellier. And I had tried to stay for a 3rd semester in Montpellier, doing this as an external project over Groningen. I actually tried. I talked to Franjo if that would be possible to do it in Montpellier the same way people do Harvard, but that didn't work out. So I went to Oxford for the last semester over Groningen. And when I finished meme, I joined I came to the beautiful city of Zurich, where I did my PhD at the Institute of Evolutionary Medicine. So just to give you an example of what evolutionary medicine is, for those of you who you've all heard about it, but just a small overview. Evolutionary medicine is trying to apply evolutionary thinking on the theory of evolution to solve medical problems. Some classical examples are cancer evolution. Cancer is a very evolutionary process. Cancer cells have some kind of variation within themselves, and they compete against each other to multiply and take over the population. And they evolve. Their high capacity to evolve is one reason why therapy fails a lot of times in cancer. So cancer is a very evolutionary process. A lot of people doing cancer research apply evolutionary principles all the time without actively thinking that this is evolution. Another classical example is antibiotic resistance. As cancer, bacteria always evolve to overcome the therapies we have. They're actually under very strong selection most of the time. And a lot of people argue that applying evolutionary theory could be one possible way to overcome antibiotic resistance evolution. So something like trying to reduce the use of antibiotics to control the infection without applying such a strong selective force on bacteria to evolve resistance, and try to wait until the body eradicates the infection, or trying to apply multiple therapies at the same time to reduce the probability of resistance evolution. Something like combination therapy for HIV infections, where they use 3 medicaments at the same time, and then the probability of evolving a mutation that's resistant to the 3 of them is very unlikely. Another example is mismatch and mismatch problems. These are problems that are a result of the modern environment being very different to the environment where we have evolved. So many people talk about carpal tunnel syndrome or low back pain, or a lot of psychological disorders as a result of this mismatch problem. Just so the projects I've done during MEME and during my PhD, in Montpellier, I actually worked with Samuel Allison first on the evolution of virulence in Ebola. That was a theoretical modeling work. I then worked with Michael Hochberg on phage therapy and using phages, which is also another evolutionary solution to antibiotic resistance. So using phages that kill bacteria instead of antibiotics or in combination with antibiotics. In Oxford, I worked in the MacLean lab on antibiotic resistance. And then in Zurich, I did mainly theoretical work modeling the evolution of immune responses. So I will be talking today about two aspects of the evolution of immune responses. One of them is the evolution of immune sensitivity and how evolution regulates the optimal immune sensitivity of a host. And the other topic is sex differences in immunity. Why do the two sexes evolve to have different immune responses? And I will explain that when I come to it. So the first part is evolutionary regulation of immune sensitivity. Just to think of how evolution regulates immune sensitivity, we have to think about the costs of immunity. So what are the costs of immunity? People have talked about two types of costs. One of them are resource-related costs, that are all the resources a host has to invest in establishing and maintaining an immune system and mounting immune responses. That these resources could have been invested in survival or reproduction otherwise. The other kind of cost is the immune damage to self. The immune responses cannot be perfectly precise, and hosts do always do some damage to themselves in the course of an infection. And actually, a lot of the early work on immune evolution, like the work of Berenberg and none other than our Franjo, was about the resource-related costs of immunity. So these papers, you can actually see it from the title, The Evolutionary Economics of Immunity, for example. These papers focused on the trade-off between how much resources go into immunity versus other aspects of survival. And they actually classify immune costs into two types. One is facultative, which are immune costs that apply only during an infection, versus constitutive, which are immune costs that the host puts into having an immune system from the beginning. So they are not related to having an infection. But actually, the immune damage to self has been gaining more attention lately from the work, from the work of Kressler and Day, for example, where they modeled the immunopathology or the damage the host does to itself, and the work of Metcalf on the trade-offs between sensitivity and specificity of immunity. And I'll just give you a short summary of the framework of these papers. So, for example, the Metcalf paper says that there are two distributions that are very important to immune responses. These are the molecular signatures that the host immune system uses to identify and detect and eradicate parasites. So if you imagine them as two normal distributions, you have the distribution of the host cells and antigens on the left side, and the distribution of the parasite molecular signatures on the right-hand side. And parasites are under selection to avoid detection. So they try to resemble cells as much as possible. Thus, the overlap you see in the middle. And the immune system has to kind of find that threshold somewhere in between to decide, okay, where should I attack and where not? And so the evolutionary result of this process depends on how much overlap there is between parasites and hosts, and what costs are related to infection, to undetected infection, to autoimmunity, to attacking cells, and so far. So this was one of the first papers to build on this framework. And that's when I started my work, and I said, well, there are two components missing in this framework. One of them is differentiating what kind of autoimmunity or what kind of self-damage that the immune system does. And the other one is some kind of ecological feedback between infection and parasites. So this means, so there are two kinds of immune damage the immune system does to self when it makes mistakes, discrimination mistakes. One of them is called immunopathology, and the other one I will call autoimmunity. Immunopathology is all the kind of collateral damage or friendly fire the immune system does during an infection. So this happens only in the course of an infection, and it's just a natural result that fever or inflammation always do damage to self. And it's an unavoidable cost of resisting an infection. And this is usually characteristic of innate immune responses, like fever and inflammation, because they cannot be perfectly precise. So you can think of it always as a kind of collateral damage that's related. So any offense has this kind of collateral damage cost, while autoimmunity is something different. It's not about doing collateral damage. It's actually about identifying self as a parasite. So it's a wrong target identity. And it's not necessarily related to infection. People do develop autoimmune diseases without having an infection. So you think of diabetes type 1, that's when the body attacks beta cells in the pancreas. So it does damage to cells without having an infection. It tries to eradicate. And it's actually characteristic of adaptive immune responses. The adaptive immune responses are antibodies that are that when directed against parasites do very little damage to self because they're very precise. But when you have antibodies against a component of self, that's when you develop an autoimmune disease. So it's a wrong target problem. The other thing that was missing from the immune evolution papers was like the sensitivity, specificity, evolution was epidemiological feedback, which means that, so if you have a population of susceptible and infected hosts, you have two processes taking place here. One of them is susceptible becoming infected, and the other one is infected individuals recovering and going back to be susceptible. And epidemiological feedbacks mean that, well, the optimal immunity a host has depends on how likely it is to become infected or to recover again. So it has a lot to do with how many individuals are infected. And this has to do with the immune strategies of all other individuals in the population. So a lot of papers have shown that you have to take an adaptive dynamics framework to arrive at the endpoints of this evolutionary process. And you cannot just focus on the individual. So taking epidemiological feedbacks into consideration can change the prediction. So you always have to take this into consideration, which was missing from the Metcalf paper. So actually, that's where I picked up on this topic, and I constructed a model on the optimal immune sensitivity, taking into consideration the two types of costs, of self-damage costs and epidemiological costs, feedbacks. And I tried to answer these questions. So first, what are the endpoints of immune sensitivity evolution of innate versus adaptive immune responses? And what are the effects of pathogen virulence, host lifespan, and immune memory, with the fact whether you get back to being susceptible after recovering from infection or being immune for lifelong. So I constructed some mathematical models, some classical SIR models of disease transmission, where the host can have an immune strategy, an immune sensitivity strategy, and the higher the immune sensitivity, the more rapid they would recover from an infection. And there were two kind of costs for high sensitivity in this model. One of them is increased collateral damage, which means only those who are infected would suffer from this increased collateral damage if they have high immune sensitivity or increased autoimmunity, which means those who would recover very quickly have a lifelong higher risk of developing an autoimmune disease at a certain point. I will not bore you with the equations and the details of the model. I will jump directly into the results, but I have them in extra slides if you want to see. So these are some of the results I got when I looked at innate immune responses. So only looking at the collateral damage immunity does. First, I looked at the effect of the virulence of the pathogen. So on this graph, you can go from very low virulence. You can think of something like a fungal infection that would never kill its host, but it's still unpleasant, all the way up to a very deadly virus that is very likely to kill its host. And what you see here is that whether or not the host has immune memory, it will always evolve a higher level of immune sensitivity the more virulent the pathogen is. And this makes sense for me because it makes no sense to try to tolerate deadly pathogens that are likely to kill you anyway. So probably the deadlier the pathogen, the more you try to get rid of it, even if you do a lot of damage to yourself. But if the pathogen is very benign, probably it's not so bad to kind of evolve very low immune sensitivity and tolerate it. If jumping to the effect of the lifespan of the host, so I have here very low natural mortality, long lifespan on the left, going to very high natural mortality on the right. And here we see that actually the results depend on whether or not there is immune memory. So in the absence of immune memory, that's the dashed line, there's a slight increase with the optimal immune sensitivity if the lifespan is shorter. And probably this has to do with with accumulating damage. So because if you don't have immune memory, and the longer you live, the more you will encounter the same infection again and again and again. So if you live long, probably you would like to decrease the damage you do to yourself each time you encounter the same infection, because otherwise you will accumulate a lot of damage. While if you're likely to die anyways very soon and never see this pathogen again, probably you could afford to have a bit higher immune sensitivity. But once you add the effective immune memory, you see that there's a very obvious decrease in the optimal immune sensitivity with a higher natural mortality, because if you're going to live very long, and once you recover from this disease, you will never get it again. So you have more benefit actually from recovering from it, even if you do a lot of damage once to yourself, but you will be immune for a lifetime and you will never have to deal with this pathogen again. So longer lifespan would select in this scenario for a higher immune sensitivity. Jumping to adaptive immune responses. So this is when the cost of having high immune sensitivity is a higher risk for developing autoimmune diseases for the whole life. And looking again at the effect of the pathogen virulence, we see a slightly different pattern. So if the pathogen has a very low virulence, it doesn't make sense to try to evolve immune responses against it. That's what the dashed line on the left side of the figure. And then there's a slight increase in the optimal sensitivity when the virulence increases, because the pathogen is more deadly and more costly, the infection is more costly. But once the virulence increases more, there's a slight drop in the optimal immune sensitivity. And I think this has to do with the probability of encountering the infection. So if the costs are lifelong and the host has to pay the cost whether or not it encounters the infection, it has to take into consideration how likely it is to encounter this infection. And if the pathogen is very virulent, it means that infected hosts would die so quickly that the infection will never be very prevalent, and therefore it will never be very likely to encounter this infection. So suddenly or not suddenly, but the optimal immune sensitivity would drop with in the higher range of virulence. If you look at the effect of natural mortality, so again, we have the dashed line for the no memory scenario. And you see that actually the higher the natural mortality, so the shorter lifespan is, the lower the optimal immune sensitivity in the absence of memory is. And I think this has to do also with the fact that, so if you think of autoimmune risk, it's a kind of a lifelong additional mortality. And the shorter your life is, the more significant this extra term of mortality would be for you as a host. And the less you can afford to actually increase your mortality rate in order to have a better immune system. So that's why when your natural mortality is very high, you would probably evolve less immune sensitivity. If you look at the solid line, the scenario when hosts have immune memory, you see the same effect on the right side. So when the higher natural mortality is, the lower optimal immune sensitivity is. But still, if natural mortality is very low, which means if the lifespan of the host is very long, you would evolve low immune sensitivity. And I think the reason is because if recovered hosts have immune memory and they live very long, they will be resistant and in the population for a very long time, again decreasing the probability, the prevalence of the disease and the probability of encountering this infection. And it's a kind of evolutionary herd immunity. So this will select for a lower immune sensitivity because it's actually very unlikely in this scenario to encounter the pathogen. So some small summary and some conclusions. Lethal pathogens select innate immune responses to react in an extreme manner. So it increases optimal immune sensitivity. And this actually might contribute to phenomena like the cytokine storm. You have probably heard about the cytokine storm in relation to COVID infections. It's a rare case where the immune response is so strong that it actually does more damage to the host than the, sorry, my phone was ringing. It does more damage to the host than the pathogen itself. So why is someone calling me right now from England? I never received this before. Okay. So it does, they're calling me again. This must be urgent, sorry. But I'm applying for some jobs, and that was the job I applied. So I really had to pick that up, but I could reschedule it for tomorrow. So we're okay. Good. So I was talking about the cytokine storm, and that is when the immune reaction does more damage to the host than the. So you have heard a lot of that. Some people die due to a COVID infection because of the cytokine storm, because they have so much cytokine that this is that they die not because of the infection itself, but because of this reaction. Charles, someone was trying to enter, but okay. And so this makes sense because so it makes sense that this kind of reaction evolves against deadly pathogens. So also, I wanted to add that selection on innate immune responses follows facultative immune cost scheme. I've shortly talked about that in the introduction, but so when looking at innate immune responses, you get very similar results to what people get when they model only facultative costs of immunity, the costs that the host has only during an infection. While when you look at adaptive immune responses, you get the same pattern or a similar pattern to what people get when modeling constitutive immune costs, and that are costs that the host encounter for their whole life. So I will jump to the second part of this presentation, which is the role of sexually transmitted infections and the evolution of sexually dimorphic immunity. And just to introduce the sexual dimorphism in immunity to you first, what I mean here by sexual dimorphism, I mean the effect of biological sex and not the effect of gender. So gender is a cultural component, which depends on how you define the two sexes, and how it's a more of a behavioral component, while biological sex is a more of a hormonal genetic component. And sex differences in immunity have been actually described in many species, from lizards to fruit flies to sea urchins to humans. And they typically describe lower immune competence in males. And I will give you some examples later on. So just to highlight how important these differences are. So until 1977, the FDA guidelines excluded women of childbearing potential from drug trials, because they back then they considered that women are too labile, and that the hormones might be too confounding to study the effects of drugs. But actually, in the 1990s, the FDA and the NIH recommended that clinical trials should include female subjects, and that was partially supported by the recognition of sexual differences in immunity. Also, just to give you a very recent example, so there are a lot of studies out there that highlight that actually men are more likely to die due to a COVID infection than females, which has been described since long in the evolutionary medicine literature. I give you a short quote. Being male is now the single largest demographic risk factor for early mortality in developed countries. This is from a 2006 paper in evolutionary medicine. So I will describe some of the sexual differences in immunity in humans. So first example is infectious diseases. Men actually have a higher mortality due to most infectious diseases. That includes influenza and COVID and a lot of others. Vaccines, actually, women have higher antibody levels after getting a lot of vaccines. This has been documented against influenza. I have one recent paper that I have published is what we looked at the sex difference in response to the HPV vaccine, the human papilloma virus, and women did have significantly higher antibody levels after getting the third dose. And malignancies, actually, all non-reproductive cancers are more common in men than in women, with very few exceptions that include the gallbladder cancer and the cancer of the thyroid. And autoimmune diseases. This is a very actually big difference. This figure, you see actually the most common 10 autoimmune diseases in a human. Pink bars are the incidence in females, blue bars are the incidence in males. And you can see that actually there is a very strong female dominance in the incidence of autoimmune diseases. Actually, almost all of them are more common in women, except for the last two that are almost equally common in men and women. So this is a very stark difference, a very interesting difference to study. Why are autoimmune diseases more common in women? And there have been a lot of theories, evolutionary theories, to explain why this has evolved to be so. Some of the earliest work comes from Marlene Zuk, who actually used life history theory to explain sex differences in immunity. And in summary, what she says is that, so the host allocates resources to different functions to increase fitness. And since female reproduction rate has an upper limit, males allocate more to reproduction and less to survival. So she says there is theoretically no upper limit to the the reproductive success or the reproductive rate of a male. So it makes sense that males would allocate more to reproduction than do females. So they will have less left for functions like immunity, and that's why males should evolve a lower immune competence compared to females. And I quote this, she said, like, males live hard, die young. And actually, Hannah Coco and Andrew Steur did one of the first first modeling papers built on this theory. So they wanted to test the work of Zuk And they challenged the assumption that immunity affects survival only, which was the main argument of that paper. So what they said, actually, immunity does not affect survival only. Immunity affects the general condition of the host as well, which might be an important factor in attractivity. So attractiveness can depend on the general condition, which depends on immunity, which means males do not necessarily invest less in immunity because they have to invest more in reproduction. They still found actually that sexual selection decreases the male immune investment relative to females, but when the dependency of the general condition on immunity is very strong, males do end up investing more resources in immunity than females. Metcalf did also a model on the trade-off between immune sensitivity and specificity, looking at or considering sexual dimorphism as well. So they employed a trade-off between the discrimination between pathogens and cells. They found that females evolve higher sensitivity when assuming that they have greater infection risk during pregnancy, and they can transfer antibodies to their offspring. So only when they took these two conditions into consideration did they arrive at the empirical at results that match the empirical results that females evolve higher immune sensitivity. Otherwise, that was not the case in their paper. Also, a very interesting paper that came 2 years ago looked at the effect of sexually transmitted infections with a very interesting hypothesis. So they said actually that females infected with a fertility-limiting STI invest more in offspring. They call this terminal investment. So if females are likely to become sterile when they get a sexually transmitted infection, they are more likely to invest more in their current offspring because they will have less chances to have other offspring later on. So for males, it would be better to mate with an infected female because these are the ones that will invest more in the current offspring and not save for the future. So that's why they conclude that actually males might reduce their immune competence to infect their mate, because for them this is the best security that their mate is infected and that they get more from this reproductive cycle. Just to tell you why sexually transmitted infections are very relevant to immune sensitivity in humans. So first of all, most sexually transmitted infections are fertility limiting. This has been well studied. They don't have high virulence in terms of mortality, but they can affect fertility. And sterility after a sexually transmitted infection is a product of the direct damage by the infection itself and by tissue scarring due to the inflammation and the immune response against an infection. And actually, due to anatomical, physiological, and behavioral reasons, susceptibilities and outcomes of sexually transmitted infections differ between the sexes. And I'll give you two examples. First, the risk of genital herpes transmission from a male to female partner is almost 4 times as much as the risk of transmission from males to females upon one contact. So the sexually transmitted infections seem to go more easily in this direction. After a single episode of sexual intercourse, a woman has 60 to 90% chance of contracting gonorrhea from her infected partner, whereas the risk of transmission for a man from a woman is 20 to 30% when the woman is the infected partner. So there is significant difference in the risk of getting an STI after contact. This is a fact sheet from the CDC, the Center of Disease Control. I would like to highlight a couple of points. One of them is they say a woman's anatomy can place her at a unique risk for STD infection compared to men. And women may not see symptoms as easily as men. And actually, STDs can lead to serious health complications that affect women's future reproductive success. So they say that it is more likely for women to develop pelvic inflammatory disease after an STD, which is a very high risk factor for infertility and ectopic pregnancy. So these are all the empirical data that support this assumption that we should take into consideration the differences in the risk and outcome of STIs when studying the evolution of immune sensitivity in the two sexes. So we constructed a model of disease transmission to see how the two sexes react to changes in the susceptibility, virulence, and immunopathology of sexually transmitted infections. We consider two kinds of differences, intrinsic and extrinsic. Intrinsic differences are a result of the fact that females invest more in the provision for offspring, while males' reproductive investment increases his mating success. So this is a very common assumption on papers on sexual reproduction, that males invest more to have more access to females, while females' investment increases the survival of their offspring. And the extrinsic differences are the differences I've introduced in the susceptibility and sterility rate of sexually transmitted infections between the two sexes. I'll put some results here. So first is I had a model where I ignored all the intrinsic and extrinsic differences just to see the neutral effect of different factors on the immune response to sexually transmitted infections. So first, I looked at the effect of susceptibility, and you see here that the higher susceptibility is, the lower optimal immune responses against the infection would be. So it means something like, if you are very susceptible and you're likely to get the infection, you would probably try to reduce your immune response because you will encounter this infection a lot, and you don't want to do, you want to do as little tissue scarring to yourself as possible. So that's when the host would evolve lower immune sensitivity in response to increased susceptibility. If you look at the sterilizing effect of the pathogen itself, how much direct damage it does to tissues, the higher the damage it does, the higher the optimal immune response should be, and the higher the level of immune pathology associated with a certain infection, which is again related to how much overlap there is between pathogens and cells, the lower immune sensitivity should be. And then I jump to the scenario where I include the intrinsic and extrinsic differences in this same model. So here you see the scenario when there are only intrinsic differences between the two sexes. This means females invest to increase the survival of the offspring, males invest more in reproduction to increase their reproductive success, and the sterilizing effect of the pathogen is similar in the two sexes. And you see that, so males are the dashed line, females are the solid line, and you see that immune competence is on the right. So females have a slightly higher immune competence, while they have a slightly higher reproductive investment than do males. No, it's the other way around. Females is the dashed line. So females have a higher immune competence, and males have a higher reproductive investment. But once I factor in the fact that the sterilizing effect of an infection can be higher in females, so this is when that sterilizing effect is 2 times as high in females compared to males, you see actually that the sex difference is even bigger in immune competence. That's again the figure on the bottom right. So because females are more likely to or are more damaged by the infection, they tend to evolve higher immune competence. The other scenario here is the effect of the direct damage, and when the susceptibility to infection differs between the two sexes. So again, this is here, the two sexes, we assume that the two sexes first have the same susceptibility. Direct damage increases optimal immune competence. And you see that actually there's very little difference between the two sexes, but still there are some differences. But once we factor in that the females can be more susceptible to the sexually transmitted infection, you see that actually males suddenly evolve higher immune competence, and females tend then, because they're very susceptible, they tend to evolve more tolerance to the infection and lower immune competence. So some short summary of the results. Intrinsic differences can select for sex-specific immune competence without considering extrinsic differences. And extrinsic differences may either augment or mask the effects of intrinsic differences depending on their magnitude and direction. So if the effect of both intrinsic and extrinsic differences are in the same direction, we expect the extrinsic differences to augment the result. And while if they counteract each other, they would probably mask each other. And the observed sexual dimorphism in immunity should correspond to the sum of both the effects of intrinsic and extrinsic differences. And this might actually explain why the differences are not universal and not as universal and pervasive across animal species as had been thought before. This is actually a recent review on sexual dimorphism in immunity across almost all animals, or at least all the animals they had data for. And what they found is that females exhibit a significantly stronger immune response than do males, but the effect size is relatively small. And that actually their findings challenge the notion of pervasive bias towards female bias immunity. With this, I come to the end of this presentation. Thank you all. Thanks Charles for organizing this, and thank you all for listening, and thanks everyone in NEEM and University of Zurich for their support.
Nice. That was very interesting. Did you get the interview for the job?
Yes. I don't know what I got. We had to reschedule a call for tomorrow. So
well, it's better than no call. So
yeah, sure. So, that's good. Just like not perfect timing, but still better than nothing.
Good news.
Good news.
I have a quick question about the first part of your presentation, where there was the difference between the lifespans.
Mm-hmm.
I would imagine, and so like that modeling, I assume is based off of like an entire population having the same lifespan,
mm-hmm.
right? So like, but like I guess I just wonder,
mm-hmm.
like for like cross-species infections, cross-species viruses or whatever,
mm-hmm.
they're going across different lifespans. So you could potentially model that, right, with like a different ratio like you did for the male-female, and see how that would change.
Mm-hmm. Mm-hmm. Yeah, that's a very interesting comment. So this model actually was a very host-centered model, where, as you say, the evolvable component was only the immune sensitivity, and the host will have all the host population will have the same lifespan or whatever, or will be facing the same pathogen. So I didn't consider any type of co-infections of facing different pathogens with different levels of virulence at the same time, or having different rates of natural mortality in the host population. But yeah, I cannot imagine now how this would change the results. It probably would.
Yeah, like COVID, right? Like minks can get it, we now know. And minks have a different lifespan than we do. But anyways, Vlad has a question.
Yes. I don't see it.
Vladimir, do you have a question? All right.
Okay.
I guess.
Just force unmute him to hear what he's saying. Probably talking to someone or something. Yeah. Lorena?
Wait, Vlad said something about I'm talking, but we cannot hear you.
I cannot hear him. Vladimir, you were mute, so
apparently he's not muted, but
yeah, not now, but he was muted like a couple of seconds ago.
Oh, he said that his mic doesn't work.
I'm talking. I think my mic doesn't work. Okay, so he's typing the question. At least you can hear us.
Okay, so I
Okay.
guess Vlad is going to type his question, and Laura,
Okay.
you can go.
And okay, so maybe Lorena, you can ask your question while Vladimir types his question.
Yes. Yeah, I arrived super late. I'm sorry. I'm really sad about it because I think the topic is really interesting. I was just wondering, this may go a bit against what you were saying, but I was wondering if similar or this effect on between the sexes is related to being a male versus being a heterozygous for the sex chromosome. So in other words, I was wondering if the same pattern was seen in the ZW systems. Also butterflies. If the same effect seen. I guess it's related to the very last.
Yes. Okay. Actually, so as much as I remember from this review, because this is where they compared all animal species, so including insects and birds that have different systems of sex determination. And interestingly, so the biggest difference they found was in insects who have a very different system for sex determination, and also who don't have testosterone. And before this review, everyone had thought that testosterone, because we know that testosterone has immunosuppressive effects, everyone had thought that this is the main culprit in this sex difference. And this is where they thought, well, no, insects don't have testosterone, but still they show the biggest sex difference in immune responses. We know in humans that the X chromosome also plays an important role because there are a lot of immune regulatory genes on the X chromosome. And just the fact that the females have 2 copies of this, even though one copy should be silent, but usually it's not the same copy in all of the cells. So some immune cells have 1 copy while the other immune cells have the other copy. And that's why they have a bigger repertoire of immune genes in females compared to males. But still, for me, all of these effects are kind of proximate effects that should mediate the effect of selection. So whether it is testosterone or the X chromosome that's mediating the sexual difference in immunity, still the optimal point or endpoint of immune evolution should be kind of independent of what mechanisms underlie this difference.
I guess it depends on what's the focal part, right? Like if it's about having the different components, as you say, in the heterogametic sex chromosome, then the females would be the ones that are compromised in their immune system, I guess.
Yes, right.
But in birds, right? For example, or butterflies or whatever.
Where the males have 2 copies of this chromosome, and that's why they should have a bigger repertoire of immune genes, right?
Yeah. I guess if that matters, then you would see it inverted. But, maybe you don't, because maybe that wasn't mentioned in the review.
I'm not sure. I should look back at the review. I know that in birds also, I mean, the effect of the testosterone is very strong, and like males who have more testosterone, you tend to have weaker immune responses. One of the memes did the study on this. It's Diana from our cohort. So in her study, she found that when a female gives birth to males in an environment with a low risk of infection, they tend to have higher levels of testosterone. So the females can kind of control how much testosterone their male offspring have. So that's a kind of a gambit, because they say, well, the risk of infection is very low. It's better to have a son with low immune competence, but very attractive son with high testosterone, kind of. And while if the environment is very infective, if there are a lot of infections in this environment, they tend to lower the testosterone these sons have, because they will need a very strong immune competence. So I think you are right. There are a lot of factors going on together here, whether that males have 2 copies of the sex chromosome or the females, and how much role the hormones play. I'm not an expert in this difference between species. But you're absolutely right. I think I should kind of look more into this.
Oh, super cool. Thank you.
Okay, so I have Vladimir's question. Thanks for a great presentation. It's very interesting. I was wondering how much of the differences between sexes can be explained by the pregnancy. Something similar to Lorena's question. Maybe there's bigger immune response of mothers to male offspring as they are more different than female body, not only due to sex chromosomes, but due to huge list of the phenotype that might cause immune responses. And maybe it's beneficial for males to have lower immune response in order to allow their mothers to survive pregnancy and their own survival. Very interesting and very like many points. Oh no, so pregnancy is actually, there's a hypothesis about the role of pregnancy in sex differences that I have not mentioned in this presentation, where there are two components. So one of them is that pregnancy itself is an immune challenge to the female body, because the fetus is a foreign body that the immune system has kind of to detect, but should not eradicate. And this plays a role. Actually, this could be, so one of the topics I will work on during my postdoc, not the current postdoc, but if I take a second postdoc in Freiburg, should be the effect of pregnancy. And how, so these two components. First, there is kind of parent offspring conflict going on here, because the placenta tends to exploit the uterus. So the female has kind of to keep this in check, but not so strong that this damages the fetus. So there is here a kind of evolutionary conflict. And actually this, I think that when the fetus is male, there is higher tendency for the placenta to overexploit the uterus. The second thing is the transgenerational immunity. So the fact that the antibodies, Diana is trying to join, that the antibodies can be transferred from the mother to the fetus, but not in males. That is the first thing. The second thing, only adaptive immune responses can be transferred. You can transfer antibodies, but you cannot transfer inflammation and fever. So also this kind of, there are two different schemes of selection on these different things when you take pregnancy into consideration. I don't have any answers to these questions, but they are very interesting.
I think Giannis has a question.
Yeah.
I actually have three.
Oh,
the first two are on the first part of your talk. So the first is that I think in the slides that you showed, the natural mortality is always lower than the parasite-induced mortality. And so I was wondering whether if you increase the natural mortality, the pattern could change. Did you look at that?
I remember that I have actually, like, the only restriction I had on this model is not to increase mortality so much that the infection does not spread anymore, like not so to exclude infection-free scenarios. But still, so in the parameters I've considered, I think you're right. I always had lower natural mortality than infection-induced mortality. But still, I'm curious, how did you find out from the figures? Like, what was the clue?
Well, it's on the figures. The virulence.
Yes. Yes. Right. I write the virulence 4.
Right.
Yes. Exactly.
Okay. The x-axis is natural mortality.
Yes. For me, it made more sense because like all of the examples I thought of are like how can it, so if you are more likely to die naturally than to die due to an infection. So your lifespan should be very short and the infection should be kind of very benign. I don't remember how I did this actually. Yeah.
I was just curious. I mean, it's not a big thing. I think it's just,
yes.
I was wondering if you looked at it, perhaps it's worth checking.
Probably I did, because I have, yes, this could be, because here I kind of tested all the ranges of both. Wait, here, no, no. This is virulence versus immune mortality. And here I have natural mortality versus autoimmune mortality. So you're right, I kind of need still a figure with natural mortality compared to virulence. But I'm just trying to guess, how it would be. It's probably, I'm not sure if this is kind of is possible, because I don't know if the natural mortality is so high, it could be that the infection could never spread. But I'm not sure if this is the case. I should go back to the script and try to play with the parameters and see if this is possible. Yes.
Yeah. So the other question was actually on the same slide as the first one, not these plots.
Yes.
So I'm probably misunderstand. So I'm trying to understand why if there is memory, the immune response is more sensitive than if there is no memory, and perhaps it's related to. It has to do with the fact that perhaps I don't understand what memory implies, or perhaps I don't understand what sensitivity implies. Because intuitively, I would think that if a host can remember an infection, well, if a host does not remember an infection, and the infection has high virulence, then the host has to mount very rapidly an immune response or suffer the virulence. While I would expect that memory to mean that the host is more ready in a way to reply, and therefore the rate of reaction should not be as fast. But perhaps I missed something on what the sensitivity means, or on what memory or no memory mean. So if you can explain this.
I will try. So first, virulence is how likely here in this model, how quickly the host dies when it's infected. And sensitivity here is two things. First, of how quickly it recovers. So if it's highly sensitive, it will just get quickly out of this infected compartment. But the cost here, because we're talking about innate responses, it would be higher mortality only when it's infected. So it means that there are two. The effect of memory is here a bit subtle. So it means even if you get the infection, and if you have high immune sensitivity, you will do a lot of damage to yourself, and that's the cost of it, right? But if you have immune memory, it means once you recover, you go to a recovered compartment, and you will never get this infection again. You are completely resistant. So what this means is that also is that the host has the host, that it will only suffer the cost of this high sensitivity if it's infected. But if it has immune memory, it means it doesn't, whether it is in the susceptible compartment or in the removed compartment, it doesn't care anymore about any cost of immunity. It only enjoys the benefit.
Okay.
And I think that's why in this scenario, the immune sensitivity is always higher if you have immune memory. This was kind of a solid result, because this is like one of the only results I could also show analytically. So if you look at the expression for optimal immune sensitivity in the presence or absence of immune memory, there is always, it's the same term minus something if it doesn't have immune memory, which means it's always less in the absence of immune memory. So it was a very solid result. And I think it has to do because here you only have the cost of immunity if you are infected. And if you develop immunity after the infection, you will never have the cost again. So like the relative benefit to the cost is much higher if you have immune memory.
Yeah. So if I say that it results from selection on spending less time in the infected department, am I correct?
Yes. I think so, yes.
Great. So can I ask the third question? Yeah.
Go on.
So it's on the second part of the talk. So it's on when you look at the effect of susceptibility on allocation to immunity, I think.
This one.
So I think it's the first results.
Yeah. Okay. When we ignore 6 differences. Yes.
Is that it? Yeah, this one. So this one shows the one on the left shows that sensitivity goes down as susceptibility goes up, which to me is a bit counterintuitive, because I would expect that if a host is likely to get infected, that's when it actually needs an immune system. If you're never going to get infected, then you can very well do without.
Yes.
So I guess I'm missing something.
Yes. Which is exactly so. You only look here. You only talked about the benefit of having this immunity, but you also should. What we considered here is that when you have higher immunity against this infection, you are also doing more damage to your cells in the course of this reaction. So if you are more likely to get the infection again, actually at the extreme part, when you go to the far right of this figure, the host is almost always infected. So first, it doesn't help a lot to get rid of the infection anymore, because you will get it quickly again. And second, during the whole time when you have this infection, you will be doing a lot of damage to your cells. So that's why the importance of getting rid of the infection decreases if you are very likely to get the infection again and again and again.
So in this model, you never recover for life.
So if susceptibility is very high, you almost everyone remains in the infected compartment, because here I didn't have any kind of immune memory in the second model. So when you're so susceptible, you get back to almost the whole time you will be infected. So that's where the relative benefit of immunity decreases.
But wouldn't it be interesting to add immunity just to see?
Immune memory.
Sorry, yes. Thank you.
Yes, this probably would change the effect of susceptibility, you're right. But the thing is, from empirical data, most of sexually transmitted infections do not the host does not develop immunity to immune memory against them. So most of them are recurrent infections. And the second thing is that because this was the first kind of try to model this effect, so I wanted to try the simplest kind of scenario to look at at things first. And I wanted to consider more complicated scenarios later on, but still for me, for a first try, it made sense to keep it simple, and also because these infections are very recurrent, so it made more sense to do an SIS model without immune memory.
Okay, thank you, Lafi. It was very
Thank
interesting.
You. Thank you.
Good luck for the interview.
Thank you.
All right. Well, if nobody has any questions, I have a meeting